Welcome, everyone. Thank you for joining Continuous Learning with BioGuard. It is my pleasure to introduce our webinar speaker, Dr. Chen Xilin. Dr. Lin got her DVN degree from National Taiwan University and her PhD from College of Biological Science and Technology at National Jiao Tong University. She's a professor in the Department of Veterinary Medicine and director of Zoonosis Research Center at National Taiwan University. Her specialties include veterinary clinical microbiology, clinical pathology, and animal cancer biology and therapeutic development. As we know, bacterial culture and antimicrobial susceptibility testing remain essential in veterinary diagnostics, though conventional workflows are often slow and species dependent. In this lecture, Dr. Lin will explore recent advances that improve both speed and clinical relevance, as well as the use of diagnostic data in antimicrobial stewardship to address the growing challenge of AMR in veterinary medicine. Without further ado, please join me in welcoming Dr. Lin.
Hi, everyone. Thank you for the introduction from Dr. Luo, and I'm Chen Xilin from National Taiwan University. So it's my pleasure here tonight for sharing the lecture about bacterial culture, how to use this kind of techniques from culture bacteria and to test the drug sensitivity of these microbes, and then we can find out the solution to treat specific kinds of infectious disease. So now I will start to share my slide. If you have much more interest about today's topics or some related products from BioGuard, the cooperation company, you can scan this QR code to follow them, to follow BioGuard in Facebook or LinkedIn. Why is so important about the microbial culture? Because right now there are so many infectious diseases, and this kind of infectious disease, if we want to identify or we want to make some kind of differential diagnosis, we need effective tools for this. So one of them is about the culture, bacterial culture, and followed by the susceptibility testing. It can prove some kind of evidence and to help us to make the final diagnosis in clinical work. And because the susceptibility test will identify the specific antibiotics that will help us if we face some kind of bacterial disease, and we don't want to use antibiotics just based on our experiences or based on what kind of drugs we own in the hospital, because the misuse of this antimicrobial drugs will cause the following up possible antimicrobial resistance. And this kind of drug resistance actually it will result in possible bad outcome when it will give us the bad results, no matter if the dog or the human beings. Because this kind of antimicrobial resistance maybe finally will give us no drug to use if we suffer from potential following infection. So basically, in clinical works, for the dogs or cats, we can face different kind of infectious disease. And most of the cases would be for example, pyoderma, this kind of skin infection, or otitis, for the ear infection, or urinary tract infection, or respiratory system infections. So when we face this kind of diseases, we need to do bacterial culture and antimicrobial test, susceptibility test. Right now, there are several bacteria that will cause the infection of, such as we've already mentioned, skin infection, ear infection, or maybe pyometra, urinary tract, and respiratory system infection. And among these bacteria, maybe we need to pay attention on specific kind of these microbes. For example, this is it. This one here, we show this, the Extended-Spectrum Beta-Lactamase, in short, ESBL-Producing Bacteria. In the investigation from our teaching hospital, we found that ESBL prevalence is around 13% in clinical isolates. And all these cases are obtained basically from dog and cat cases. So this ESBL bacteria, including E. coli, Klebsiella, Enterobacter, Acinetobacter, and Serratia. So if we focus on their genotypes, there will be, for example, TM, CTXM genotypes. And among the genotypes, CTXM1 group is the most common bacteria that we will found. So, so we will see several species. So what is the most dangerous species, or most species we will find or identified is E. coli. This is E. coli ST457 strain. So in addition to the strain we've mentioned, in Taiwan, E. coli ST131 is the second most common ESBL producing E. coli clone. And this strain could be isolated and associated with both pets and humans, so suggesting potential interspecies transmissions. So it means that if the infection occurs in dogs, cats, or humans, no matter, it can cause the partner. For example, if the dog they suffer from this E. coli infection, and if the case is not get well controlled, it could be transmitted to the human beings who care about the dog cases. So it's an important issue that we should understand what kind of bacteria right now has caused this kind of infection, as well as we need to know what kind of specific drug could treat this bacteria infection. And we use the antibiotics precisely. And then it will make sure that this drug can kill the E. coli, and we don't do misuse or misconduct of the drug usage. So for the antimicrobial resistance, actually right now as the trend among the worldwide, I think maybe for the human doctor, they are care about and they claim one of the major reasons is about not properly usage of antibiotics from the veterinarians. But from our perspective, I mean from the perspective of veterinarians, that actually I think most of us, for example, in Taiwan, when we face the when we need to do the diagnosis of the microbial infection disease, we always will perform bacterial culture and the following antimicrobial susceptibility test. So we base on the result and we choose the exact one, or we have maybe 2 to 3 choices antibiotics, and then we use. So for the treatment of dog and cat and even reptile diseases, actually we don't use these antibiotics. We won't misuse of this kind of antibiotics. So we use these antibiotics actually very precisely, and we try not to use the broad spectrum antibiotics. So it's not, I don't think it's the antimicrobial resistance is not only based on the misuse of antibiotics from vet doctor. So this is the situation right now, I think, most of the world in Taiwan. So the increase of antimicrobial resistance, actually, I think it's there are multiple reasons to make antimicrobial resistance happens. So yeah, we need to do much more studies about this. So if we have if we take a look, it's just data showed. So if we this is some kind of data that we found. For example, in Northeast China, the MDR prevalence, MDR means multi-drug resistance. So it's a very bad situation that when the dogs, they are suffer from bacterial infection and there are no effective antibiotics could be used to treat the infected dogs. So this means MDR. So MDR prevalence in pet dogs actually already decreased from around 77% to 62%. So if we look at the timeline, that we've already see the decrease in trend of MDR prevalence in pet dogs. So that means it's the antimicrobial resistance is not. I think it's not the you cannot always attribute to it's misconduct, misuse of these antibiotics from veterinarians. It's I think we need to cooperate between vet and the human doctor, and we also need to take a look that the microbes we fight with actually they are also get evolved. Yeah, because microbes, they always proliferate very fast with the high incidence rate of mutation on the genomes. So they easily can gain the antimicrobial resistance by themselves. So it's kind of evolution from the microbes. So yeah, it's a but finally, in conclusion, actually, it's a very important issue that we need to pay attention on. So if we look at the working dogs, they showed the increasing resistance. And cats, they also exhibited the higher resistance levels than dogs. So it's not a dog issue, but the cat issue. I think also the human issue. Yeah, it's a. So that's why we talk about one house. If some specific species, they are suffering from this antimicrobial resistance, it will not only on this species, and it will gradually to extend to other species, dogs, cats, and then humans. So for the many types of antimicrobiotics, colistin and carbapenems, they remain largely effective. So yeah, it's the current situation that right now, though the increase of antimicrobial resistance, but if we choose colistin and if we choose carbapenems, maybe it will much more effective for treating this bacterial disease. But again, I need to emphasize that antimicrobial susceptibility test is important before you administer this kind of antibiotics. So how should we do to get the correct result of bacterial culture and the AST test? So this is the sample collection and handling procedure. So we need to start from the aseptic techniques. We need to do the proper aseptic techniques for sampling. It depends on the lesion. The lesion may be on skin, ears, or urine, or respiratory tract. So for the urine, we will need to do we need to collect the urine aseptic. So we want to, I think the best way is we need to collect the sample directly from the bladder. And after the sample collection, we need to avoid contamination and the false negative. So it depends on how we treat the samples, how we collect the sample aseptically, how we preserve the sample in correct situation and transport them safely. So for the transport considerations, we need to know the sample should be, we need to make sure the sample should be transported in right media. And we need to pay attention on what storage condition is better for the sample. And all the samples should send to the lab as soon as possible in the right media, in the right storage condition, and as soon as possible. So if the vet doctor, if we already collect the samples, and I think the most important step is we need to communicate. We need to trust, and we need to communicate the stuff from the lab, diagnostic labs, and we need to co-share the information that we've found, and the stuff from the lab or the lab doctors that will help us to get the proper culture and show us the possible AST result. So as we've mentioned before that we have the 4 stages to help us to get the correct microbiome test and the AST test. So for the key sampling size that we mentioned, basically the most infection site will be skin, ears, urine, and the respiratory system. So for the skin, before sampling, we need to clean or disinfect with, for example, alcohol. Yes, 70% alcohol, and the proper disinfection with alcohol on skin is important. And then we collect the sample from the lesions. And for the ear, we try to collect the samples with proper tools. And these proper tools, maybe we need to avoid the contact with ear canal walls. And for the urine, because we need to collect the sterile urine and not the urine that already passed from the they've already maybe in the ground collect. We cannot collect the urine and they are in the ground or that is, so it's best for us to do cystocentesis to collect the sterile urine, and it will help us to get correct result and prevent the false positive data of urine bacteria culture. For the respiratory system, basically the sample will be bronchial lavage. Use bronchial lavage or protected brushing to collect the even in some cases, the infection could be in lower respiratory tract. So bronchial lavage, our brushing, will help us to collect the right sample, and the sample may contain the possible pathogens that help us to identify it. So after we got the bacterial culture, and the next step will be the interpretation of culture results. So how can we differentiate pathogens? Differentiate pathogens with contaminants, because contaminants will give us the false positive data. So we need to do this. First, we need to. It is the very important step that at the first stage, we need to collect the sample precisely, and so we won't get the contamination sample. Also, we can use the specific cultural agar. If you could establish the good connection with the staff from diagnostic lab, we can tell them that, oh, this sample is collected from respiratory infection, and usually the staff from the diagnostic lab, they will use the specific culture plate, and this culture plate will help us to eliminate the contaminant pathogens, and they help us focus on the pathogen resulting in the diseases. And we also need to pay attention on growth pattern, because each culture we may get. I think pure growth is not normal. Basically, we can observe usually it's mixed growth. For example, in this agar plate, this different color mix different kind of bacteria. This is the common situation that when we do bacteria culture, we can observe the mixed growth. So which we need to, so the next stage is about what kind of bacteria is the major crime maker to result in the infection. So the mixed growth is a good situation that we need to think about. Well, what kind of bacteria result in the disease. And why mixed growth is the common phenomenon, because in respiratory tract, in the oral cavity, in the ear, on the skin, there are so many normal flora. This normal flora means actually we always coexist with the microbes. So even now, on the skin of healthy people, we have the normal flora. That means we have the many bacteria, and these bacteria, they are harmless to us. So they could also be cultured in the test. So for the growth pattern, maybe you can find the different kind of growth patterns. It will be interesting, but it will be also important for us to differentiate the specific pathogen who dominant in this infection. And we also need to differentiate the pathogen with normal flora. So this is kind of growth pattern that we've found. So on the other hand, negative culture is also the common findings if we perform the bacteria culture. Yeah, that means you get the culture plate and you place the sample on the culture plate, and for the 24 or 48 hours, then there is no bacteria clones found on the agar place. So this is the negative culture. So in this case, maybe, yeah, that's true. This case is not caused by bacterial infection. It's one of the possibility. Another possibility would be the false negative result. That means maybe we don't get correct samples. Maybe at the right, maybe it's not at the right site, not on the right time, or the something wrong on the transport procedure. So in this time, how to if we need to make sure that double sampling, sample the case again, is suggested. So we've already found that if we got the bacteria culture, so we found some specific bacteria, and then we'll do the susceptibility testing. And this test, we can call the AST in short term. And you can find more methods, our tools, our service in the in BioGuard website if you are interested in susceptibility testing. And this test will help us to find the specific drug to give us the indication what kind of antibiotics we can use. So there are so many methods. You can use disk diffusion method or MIC testing methods, and you will get the result sensitive resistance. Sensitive means that this bacteria is sensitive to specific drug, for example, to cefalexin. And R means resistance. So that means, oh, this bacteria is resistant to cefalexin. and I is intermediate. It means in there some kind of gray zone that yeah, they have some level of killing bacteria effects. However, it's not effective enough, for example. So yeah, it's happy to see if the bacteria is sensitivity to some antibiotics, and it's will give us it will always make us headache if we found, wow, there is no drug, there are all are without. It means the bacteria are resistant to the antibiotics that we will put into test. So yeah, this how the way we interpret the result. So the key limitation is test accuracy at the reproducibility. So as I mentioned before, it is suggested that perform the test maybe in 2 times. You sample 2 times and perform the bacterial culture and AST test for 2 times to see if this data is the same, if they are reproducible. And it will help us to increase the test accuracy. So if we see the bacteria are resistant to all the antibiotics, it will call them multi-drug resistance. And as I mentioned, the Extended-Spectrum Beta-Lactamase, the ESBL bacteria, they are the main bacterial population possess multi-drug resistance. So for AST testing, that we need to make sure these 3 points listed here. The first is about before the start of any treatment, please collect the sample before the start of any treatment with the antimicrobial drugs. So please pay attention. Please don't already treat the antibiotic drugs and you found it's not effective, and then you collect the sample and do the AST. Actually, if the case already take some antibiotics, it will affect the results. For example, maybe we cannot get any bacteria clone because these antimicrobial drugs partially kill the bacteria, so make the bacterial concentration is not enough for our culture. So do remember that all the AST or bacterial culture is performed before treating any antimicrobial drugs. So yeah, this is the first stage that. And another time point that we can perform AST is about during the treatment. Because during the treatment, if we want to monitor the effectiveness of the treatment, of course, we can observe the clinical response of the cases. But the AST will help us to make the clear decision if we want to continue our current treatment or we need to replace the current antibiotics with other drugs. So this is another very important time point that we perform the AST. So yeah, and the third point is about if the infection is not responding to our current treatment. Actually, we definitely need to do AST to give us some kind of evidence or indications for helping us to choose the maybe good antibiotics, effective antibiotics for this disease treatment. So currently for the antimicrobial susceptibility testing, there are a different kind of methods. So we have the phenotypic methods. For these methods, we can perform the bacterial culture first, and then culture, and then use the what we said about the drug disk, and to analyze the results. This is the classical methods that used. Or you can use the molecular based methods. Use PCR or qPCR. Use next generation sequencing for finding the possible bacteria within the lesion. Or we can use multi-toph mass. So it can help us correctly find the bacteria that cause the infection. And during the incubation, you can also add in different kind of antibiotics. So after the reading of the after several hours, you can get some kind of result that will help us to know which kind of antibiotics that will kill the bacteria within the samples. So, but for these methods, currently in veterinary clinics, we do phenotypic methods for the most of the time. So for the phenotypic testing, disk diffusion is the major time we used, but maybe we have there are new machines now for us to increase the accuracy and the efficiency of AST methods. So for the disk diffusion, for the MIC, minimum inhibitory concentration, it will tell us about the lowest effective antibiotic dose. So it will not only tell us about the specific antibiotic types, it can also tell us about the proper dosage that we should use. So for the result, we can find if the bacteria is sensitive or resistant to specific antibiotics, or is intermediate in the gray zone. So for the conventional AST test methods, actually it's a very labor intensive. So you may need a lot of time to get this, and you will need to use long preparation and incubation time, and there may be the high risk of cross-contamination, because during the culture, there will be some possibility for the contaminant, and so it's the major problem. Cross-contamination actually is the major problem for the conventional AST. So if we use the specific machine, for example, the AST machine provided from the BioGuard Corporation, and we can fast get the result, and we can perform the dual sampling and with the high accuracy, and it's automatic interpretation. I mean, it means that all the procedure is performed within the machine. So there will be less steps performed by human, so it will decrease the possibility of misconduct and cross-contaminate. So, for example, for this kind of machine, you can just put your sample inside, and just later you can get the result. Okay, so as the continue for the machine we were provided in the last slide. Is the reagent they provided, and it's the growth medium for the bacterial culture. So you can also you maybe need you don't need to transport your sample to other labs. You can perform. You can also send to the central lab. But if you already have this equipment in your diagnostic, in your hospital, in your clinics, you can do the bacterial culture and AST by yourself and get the result precisely. So this is we've mentioned before that how we collect the specimen used in the specific device. For example, for the sterile urine, we will use catheterization or cystocentesis for this way, for us to correct the sterile urine. And for the feces, we use swab. Skin, we use swab, ear swab. For the pus or wind or tracheal. Tracheal maybe if the mentioned to the lower bronchial infection, we need to use the lavage or brush. So if we use swab to collect the sample, it's good condition to collect enough sample. Less sample will make us found the not in maybe we culture no bacteria, but too much sometimes it is not good. So we need to collect just proper amount of the sample. So for the machine that we mentioned in Bacterial company, that they have different kind of antibiotics. So there's a panel 1, panel 2, and panel 3, and you can find out that each panel contains the different kind of the common antibiotics that we've used. And they also provide the customized panels. Depends on the need of the vet doctors. So it could be discussed to customize the specific panel belong to yourself. So this is what we said, that the data interpretation. So for the clinical decision making, based on the susceptibility results, we can see we need to correlate the lab data on what kind of bacteria we've cultured, what is the proper antibiotics we could use. We need to correlate this data with clinical size, with the infection size, and with the severity of the disease. And because the test result will also give us the suggestion about the drug dose. So the pharmacokinetics or pharmacodynamic principles for the drug and the penetration, the dose frequency is also important. If we know the specific drug may be effective, the next step, we need to know the PK or PD and to find the proper dosage for the treatment. And we also need to, during the treatment, we also could perform this AST test because it will help us about the therapy adjustment. When should we to escalate to adjust the dosage, or maybe it's time for us to change another kind of antibiotics. So it's important for if we got this culture and AST result, it could help us to use the appropriate antibiotics. So next, I will give some case study and to show the importance of AST test and bacterial culture. So for this case, it's 5-year-old spayed female Labrador with a third urinary tract infection in 6 months. Yeah, within 6 months, they have third. The dog is suffered from 3 times of UTI. So previous empirical treatment is the antibiotics is amoxicillin and the amoxicillin clavulanate. And the culture with this time, the culture result is the bacteria result in the UTI is E. coli. And this E. coli is resistant to amoxicillin and yeah, so it's obviously for the previous infection, they treat amoxicillin. But for this time, this bacteria have already harvested the resistance to amoxicillin. So right now, the data showed us its susceptibility sensitive to trimethoprim, sulfa, and enrofloxacin. So it's a very clear sample to show us that this bacterial culture and AST is important, especially for this kind of multiple infection, reoccurrence infection cases. So for the following the clinical decision, the doctor they selected the sensitive microbiotics based on the AST result, and the with the extended treatment and the courses, and then they perform the AST, the culture and AST test again to confirm the resolution. So at the end, that the diagnostic image has already, they also perform some diagnostic image. So at the end, this case is this case actually is responds well to the treatment. So in addition to cats or dogs, and right now we are in the era of exotic companion animals. I think many of you may face to now people they not just have dogs and cats with their companion animal. They also like exotic animals as their part of family. So it's for the exotic companion animal, the unique challenges will be a small sample volume. Yeah, because maybe the body size is smaller for some exotic companion animals. For example, very small turtle, or for example, hamster, or small rabbits. And limited drug safety data, because exotic animal actually they contain many kinds, many different species. So most of these exotic companion animals, there are few drug safety data on these animals. And we always use off-label use drug, right? So for the exotic companion animals, and for the antibiotic use for this kind of cases, we face these different problems that we need to know. So actually, the common pathogens cause the infection in exotic species are listed in this slide. For rabbits, we can see the Pasteurella matosida, Staphylococcus. For the rodents, Mycoplasma streptococcus will be the major pathogen resulting infection. For the birds, E. coli, chlamydia, Pseudomonas. For the reptile, Salmonella and the protease. So this is the common pathogen in exotic species. So for the sampling, sample collection tips for this exotic species, we need to use micro swab for the smaller species. And for the feces, yeah, it may be a little bit difficult, but fresh sample is always important, I think. So maybe we need to try our best, or the pet owner, they need to try their best to collect this fresh samples. And for the overall conjunctiva samples, it's we need to possess the gentle technique to avoid distress for this very small species. And minimize the restraint time is important. So proper restraint techniques and gentle movement is important for collecting the sample. So yeah, it's need to, there is also important for us to know that many antibiotics that are safe in dogs and the cats may be toxic in exotic species. So if you are already the specialties, the professionals for exotic companion animal, I think you've already know that the antibiotic, not all antibiotics is useful or could be used in exotic species. So it's important. So maybe we need to study much better for the exotic species and for the bacteria infection cases and how to use the antibiotic properly. Okay. So for the principle that we've already known that we need to based on the culture result to use the proper antibiotics, and we should try our best to choose narrow spectrum antibiotics instead of broad spectrum, because broad spectrum will result in the possible outcome of multi-drug resistance. And we need to educate the pet owners, our clients, about the complete treatment course. I think we've already known that if any people or any case they are taking antibiotics, they should complete the treatment course. They should not just take them on 2 to 3 days and the symptom relieved, and then they say, okay, that's fine. I won't let my dog or cat or rabbit and stop the antibiotics automatically. It's a very bad habit, because it will result in the recurrence of the infection. At this time, this pathogen could harvest drug resistance. So yeah, it is very important. So find the specific pathogen and antibiotics and complete the treatment course. That's very important. Yeah, and also maybe we need to take care of some possibility if they have specific drug allergies. So the patient history is important. And we need to make the very good record about every treatment, every decision making, because we could be good friend of this of our cases. So we need to document each medical records for the follow-up. Okay, so at the end of my talk today, I think this culture and the AST test is to help us enhance the decision maker to get the precise treatment. we need to get the good communication with the lab to help us, to help each other to get the correct result and the guideline, no matter for the diagnosis or the treatment. And we also know we has the responsibility to use the antibiotic correctly, because it's not just the issue on the animal we treat. It's also will affect human beings and also will affect the environment. That's what One Health concept right now. So thank you for your attention, and any questions or comments is welcome. Thank you. Okay. So I think the slide will be the slide of this lecture later will send to you from Dr. Luo. So yeah, I will share the slide with you. Okay, and from the question from the audience, have it's a very very important question that how to differentiate between normal flora and true pathogens. Yeah, for example, I think I'm not sure, but Staphylococcus aureus actually is very famous, right? It's a very important bacteria that result in pus formation. If for the skin infection, you can get the pus. Yeah. So under what situation we will get the wound infection and pus. That's because if you got some kind of wound, maybe they just kind of specific wound, maybe by cut or maybe by they fall down and got the wound, got the lesion. And the Staphylococcus aureus actually is the normal flora on the skin, on our skin. Maybe also on dogs and cats. But if we got the wound, the Staphylococcus aureus will enter from the skin to the wound and then enter into the within the blood vessel, within the wound infection. So it will go from on the surface of our body, and then the Staphylococcus aureus into our body. So this kind, Staphylococcus aureus, in the normal situation, is the normal flora. But if they got the infection, it becomes the pathogen. So yeah, sometimes it's hard to define what is normal flora and what is pathogen. But the very important principle is about normal flora. They need to reside in the normal place they already live. For example, Staphylococcus aureus, they can only live